Hi everybody, my name is Jumaili, and today I'm going to be presenting investigating gene dysregulations associated with antifungal resistance in Candida strains from Northern Ontario. Before we get started, I'd like to disclose that I have no relationships with any for-profit or not-for-profit organizations, as well as the fact that the financial support for this project is coming from NOAMA, Awesome University, NHRC, NSERC, as well as the Northern Cancer Foundation. A little bit of background information in order to get started. Oral candidiasis is an infectious disease that's caused by overgrowth of the naturally occurring candida yeasts that are found in the human microbiota. This overgrowth is most often caused by homeostatic imbalance in immunocompromised individuals, and when left untreated, oral candidiasis may even further develop into severe fungal diseases that can lead to mortality. This study utilized two different strains of Candida species known as Candida albicans in order to identify gene dysregulations that are associated with antifungal resistance, which is a prominent issue to do with treating this. The methodology for this project utilized live fungal culturing, in which Candida isolates were separated from previously obtained salivary samples from patients in Northern Ontario that had oral candidiasis. Two strains, the C. albicans resistant as well as the C. albicans susceptible, were streaked in order to then create liquid cultures for various treatment conditions. So we had control samples as well as the physiological doses of two antifungals known as fluconazole and clotrimazole. These liquid cultures were used in order to assess growth patterns of each treatment. Cell pellets were taken at various growth time points from the different liquid cultures and used for RNA extraction, which was then followed by cDNA synthesis, and then finally real-time quantitative polymerase chain reaction, also known as RT-qPCR, and this was used in order to quantify gene expression relative to two housekeeping genes found in the literature. Our growth curve results show that the resistant strain tends to grow more than the susceptible strain in all three of our treatment conditions. These results verified that our susceptible strain was truly susceptible, since both azole treatments were statistically significant from the no antifungal treatment at the mid-log growth phase, which is around 12 hours. No statistical significance was found in the resistant strain at the mid-log growth phase, which meant that both azole treatments had little to no effect on growth, verifying resistance. Moving on to our qPCR results, we see that CDR1 was significantly upregulated in the resistant strain in the fluconazole condition when compared to the susceptible strain. Since CDR1 is an ABC transporter, which stands for ATP binding cassette, this finding may explain a mechanism of drug evasion as resistance. Having more drug efflux pumps, such as CDR1, can give the cell increased resistance to drugs such as fluconazole, because there's more pumps working at once to rid the cell of the drug. UPC2 is a transcription factor that activates many genes that are directly involved in ergosterol biosynthesis, an important sterol in fungal cell stability. We found that UPC2 was upregulated in the susceptible strain as opposed to the resistant strain. We also found that SMF12 was significantly upregulated in the fluconazole condition of our resistant strain. SMF12 is a gene that is responsible for manganese transport. Manganese is an important cofactor for many enzymes and is crucial for cellular metabolism. Higher amounts of manganese transporters alludes to the resistant cells trying to find alternative ways to grow while azole drugs are present. To conclude, CDR1 and SMF12 were both found to be upregulated in the resistant strain of C. albicans, genes that are responsible for drug efflux pumps and manganese transporters, respectively. We also found that UPC2 was upregulated in the susceptible strain of C. albicans, a gene responsible for sterol biosynthesis. I'd like to thank my supervisors as well as NASM and Laurentian University for the opportunity to conduct this research.